It's my pleasure to introduce Andrew Blumberg, and the title of his talk is Statistical Foundations for Detecting Evolution of the Shape of Random Geometric Objects.
Well, thank you. Thanks for inviting me, and thanks, for coming to listen. Yes, as perhaps you can tell, there's been a bait and switch. I was going to talk about work with Abby Hickok on estimating the scalar curvature of compact Riemannian manifolds, but it occurred to me that there's no persistence in that, and maybe I should talk about something in that direction. So I want to talk about some joint work I did with a statistician named Anne van Delft on on a new foundation for doing statistics to analyze geometric time series. So let me start with the overview. We want to study the evolution of shape over time. So we're thinking of this as a process where we have point clouds that are generated by sampling from some sort of temporally indexed series of geometric objects. So we have finite samples reflecting some time-varying underlying series of metric measure spaces, something like that. And there are three kinds of questions that I want to explore about this or describe to you in the talk. One of them are foundational questions, namely just what should the statistical foundations of time series analysis for geometric data look like? Two are sort of algorithmic problems, namely, given these foundations, can we compute the relevant test statistics efficiently? And do they work do we have guarantees about their performance in non-asymptotic finite sample settings? And third, what kinds of applications is this useful for, and what might you do with this? So I want to tell you something quick about all of these. So let's move on. And sort of our main slogan, and what we're going to do is the idea is we're going to use ideas from what's called functional time series analysis as a convenient theoretical framework for studying the evolution of shape data. And as I mentioned, this is joint work with Anne van Delft. And our goal here is to integrate this kind of time series analysis, this geometric time series analysis, into this place where we can do lots of interesting statistics. Okay, so we're interested in data sampled from objects that we're assuming have potentially complicated low-dimensional geometry and topology, and where the underlying object that we're sampling from is changing over time. So some kinds of examples of this are things like, and kinds of geometric questions we'd like are cobordisms, right? Which I'll show a picture of that, but which you can think of as a manifold smoothly evolving in time. And we can think of things close to manifolds, like spaces with corners or singularities, or unions of manifolds with differing dimension, possibly where the singularity appears and disappears, or the dimension of what we see changes. And of course, many examples, and certainly from my perspective, some of the examples I'm most interested in come from genomics, and what you're looking at is probably not anything like a manifold, although it does have some notion of local metric geometry, and conjecturally via some form of the anthropomorphic principle, we hope that meaningful clinical or scientific signal is present on low-dimensional subspaces. And then just as an aside, by anthropomorphic principle, of course, people make this assumption in genomics because without it, you couldn't say anything. Okay, so these are the kinds of things, and what we're interested in, and I want to talk about the statistical problem I'm going to focus on today is very basic. And I think, there look like there are two other cool talks at least about time series analysis, I think using zigzag persistence in this conference. And we are doing something even simpler, I think, than what's happening there, in the sense that we're looking just to detect non-stationarity, to detect change in the underlying type without necessarily even locating breakpoints. But let me now give you some discussion of our framework. So we're going to encode shape data using any kind of shape descriptor that takes values in a metric space, and is sensitive to whatever kind of change we're interested in. So as everyone knows, persistent homology is a great qualitative shape descriptor that lands in a metric space and encodes information about both scale and shape, or really shape at varying scales, right, as we know. Multi-dimensional persistence works perfectly well for this purpose, and really anything else you can think of that you can put a metric on. But maybe I'll talk in terms of persistent homology to be concrete and to fix ideas as we go through. So the sort of standard view of topological time series is something like, and just this is something I said earlier in the talk, but just writing it out more formally, is that the data is a sequence of point clouds. So each x sub ti is a finite metric space, which we think of as IID samples from some underlying metric measure space. And the metric measure space, in the sort of very most classical picture of the formal framework for topological data analysis, or for the statistics of topological data analysis, you have a fixed metric measure space, and you have samples from it, and you're trying to recover something about the support or something about the support and the measure. Maybe their homology. Here, it's very similar, except we're assuming that the underlying metric measure space is potentially changing over time, or the underlying measure is changing, even if the metric space is not. And of course, the most naive thing you can do is to push this forward and obtain a sequence of barcodes from these things. Again, there are lots of cool ideas about using zigzag persistence to analyze this kind of stuff, because it's a very natural thing to think about, which I will not talk about. Well, that's not what we're doing in this talk. So what does it mean to interpret this in terms of functional time series? So the big innovation, again, that I want to advertise is that we're looking at this, that it's powerful to think of this as in terms of functional time series. And the reformulation that we're doing is that we think about a function space. So we have some function space, which denoted by C, from metric space to metric space, and I'm going to be, in order to keep this sort of manageable, given that it's a 30-minute talk, I'm going to not say so much about exactly what function space we take, but some kind of function space. And we're going to think about a stochastic process that lands in the function space. So what does that mean in terms of point clouds? How are you supposed to think about what we're doing in this language? Well, the idea is that an observed sample we think of as the image, the evaluation of a function at a bunch of fixed points, M1 to Mn. So what we see, the point cloud that we see, is the values of the function evaluated on some unknown parameters. And there are lots of ways you could think about what these unknown parameters might be. Maybe they parameterize coordinates on the manifold you're looking at. One thing to note here is that for technical reasons, and by technical reasons I mean to make the analysis tractable, in this framework we imagine that the size of the point cloud is a function of and usually the growing function of the length of the time series. And so we can think of this as sort of an upper triangular sort of thing, which I'm saying to give you a sense of what the asymptotics are supposed to look like. Okay, so this is the sort of theoretical move, which is that we're going to think about our point clouds as the images of functions, and the stochastic object that we're studying are the functions. Okay, so now I'm just saying this, the point clouds we observe, I guess I've written what I just said, but just to repeat, the point clouds we observe are the images of some function evaluated at a bunch of unknown points. And we think of those as parameterization, I shouldn't have said function, parameterization of the geometric object. The reason to do this is that this move turns out to provide a lot of tractable technical tools to describe rates and asymptotic distributions for test statistics, and very generally to analyze, there's been a lot of work recently, very successful work, on functional time series analysis. And the point of doing this here is to make use of all of that theoretical machinery in the context of topological data analysis, or more broadly, geometric data analysis. And the kind of thing that's nice about this is it gives a very good framework for talking about various kinds of dependency structures. And so, for instance, you can think about a class of locally stationary processes, which just means that in a small time they look stationary, but in larger times they're not. And there are lots of different kinds of models of non-stationarity, of course, but this turns out to give a very rich language for a tractable class of non-stationary phenomena. Okay, so how are we actually going to get test statistics out of this, and what are we going to do? The thing to observe is that TDA and geometric data analysis implicitly relies on Gromov's characterization of metric measure spaces, right? And this idea has come up. I think there's discussion of things like curvature sets, and this idea, if you think about it, pervades lots of the subject. And the point is just that a metric measure space, what Gromov proves is that in his famous chapter 3.5 of his Riemannian structures volume, is that a metric measure space is determined by the infinite dimensional distance matrix distribution, where what you look at is, you have an infinite dimensional matrix, which is produced by sampling from the points of the metric measure space according to the measure and writing down the distance. And maybe more importantly, the infinite dimensional distance matrix distribution is determined by the increasing sequence of finite dimensional distance matrix distributions. Okay, so and of course, since that's what we really have access to in life, this tells us that that's a complete invariant in principle of the kind of underlying stochastic geometry that we're studying. Now, the first observation for us is that this extends very naturally to the setting of what you might call metric measure preserving dynamical systems, where you have a tuple where you have a metric measure space here denoted S, and you have a endomorphism theta S, which is a function from S to S, which is measure preserving. And in this context, this is a model again of some sort of time series. And in this context, again, this is determined by the extended distance matrix distributions. And so this lets us use distance matrix processes as a basis for inference. So we're going to work with the push-forward processes generated by applying persistent homology to finite distance matrices at different times. And another key fact for us is that these measures can be characterized by the volumes of balls. And this is sort of a technical thing that, there's an interesting question for an arbitrary measure. Is it determined by its value on balls? And it's not always true, and there are hypotheses under which it's true. The point sort of is if you take a Kara-Theodori construction of an outer measure, you can recover the actual measure just knowing it on balls, taking the inf on covers by balls for the full measure. And the kind of hypothesis you need for something like this to be true is some kind of covering property. And so it's related to things like doubling dimension. And of course, we know that the space of barcodes has infinite doubling dimension, but there's a very nice theory, I think due to Don Sheehy, about this sort of what are called nearly doubling measure spaces. And it turns out that's kind of enough to get a characterization, under some restrictions on the barcode space we're looking at, by ball volume. So we can have, and the point of this is that we can have test statistics that are determined in terms of ball volumes. Okay, so what we look at is we consider maybe a process that is, what is the volume of a ball of radius r around our, the persistent homology of our point cloud. Right? This is a process as a function of the radius. And the kind of test we might ask for is that as r varies, and as t varies, so for all r and as t, the time parameter varies, the expectation of this is stable. That's the null hypothesis. It means it's stationary, or a proxy for it being stationary. Or the alternative is that the expectation changes as time changes at some radius r. Okay, so this is the kind of framework we can think about. And what does a test statistic for this look like? So let me write it down. What we observe are these point clouds. They're indexed by a time variable, and n of t is the size. And we can compute the persistent homology of these things. That's easy. And now we define a partial sum process, which I've written the formula here, but basically this is the sum over all pairs of an indicator about whether or not they're within R. So this is some kind of empirical estimate of ball volumes around each of the points. Okay, and this is something like a U statistic. So we have this partial sum process, and it's a function of this parameter U as well as the radius. And that U, right, what is U indexing? That's taking us across the time series, right? So we look at how the partial sums vary as we move in time. And that's going to gives us a family of test statistics that we'll use to detect non-stationarity. Now, in principle, one could, although I won't say anything about this, one can also use this kind of thing to detect breakpoints, namely where non-stationarity might occur if there are discrete changes, which makes sense in the context of a model like local stationarity, where there's sort of unchanging periods where things aren't changing or there's no dependency structure, and then suddenly there is. Okay, so if you rescale this thing by root t and look at this normalization, it says observe. Of course, this is not intended to be obvious during a talk like this, but it is true that this associated quantity under the null hypothesis has expectation 0. So a very reasonable kind of test is to look at the sup, or equivalently, the integral of this quantity, right? Natural test statistic. And the thing that you can prove is that this converges weakly to Gaussian processes, both under the null and the alternative. Different Gaussian processes, but it converges. And you can say things about the rate. I mean, that's kind of the point of this framework, is that we have lots of tools to talk about the rate at which it gets there. And so there's now we have a paper on this that's on the archive, and if you look at it, you'll see there's a bunch of sort of delicate stuff that goes into actually using this test, namely figuring out what the cutoff should be. And that requires the test has to be normalized, and the normalization depends on some kind of covariance estimates. Covariance estimation for U statistics is something that people have worked on, of course. It is not well adapted to the specific needs that we have here. And so, we had to do some work to figure out how to normalize this. One of the things that drives the difficulties here and the interest is that for our purposes, we care an enormous amount about finite sample performance for obvious reasons. Like the data sets, as I'll show you at the end in an application, the data sets that we care about are not very big. And so we're really not in the asymptotic regime. So we really need to worry about what do these things look like in finite samples? Do we have adequate statistical power to see anything? Also, these kinds of tests, if you're not careful, are very sensitive to outliers. And in some of the applications, the genomic applications that I'm interested in, outliers are a problem. And, just to say some words without writing any formulas, we have sort of some theory of sort of self-adaptive, data-adaptive weighting functions that do a little bit of smoothing in the tails to try to control the problem of outliers. Now, this is a subtle question. One could give an interesting, I think, separate talk on this, but let me just raise the point that understanding what an outlier is in a setting where you're looking for non-stationary behavior is complicated, because what looks, changes in things and things that suddenly look far away might well be a function, a sign of non-stationarity. So, I mean, that's always a problem with outlier detection, but there's sort of some slightly novel features to it in the context of time series. Okay, so we do these things and we get this test. And computing, it's very, if you look back, let's look back at the formula. Oops, where are we? Here we go. Nope. There. If you look at this, this is easy to compute, right? There are very good codes now for computing persistent homology. If we were doing multidimensional persistent homology, this would be rather more expensive. But this is relatively easy to compute. And, we can simulate to get under the null to get the quantiles for figuring out how to use the test statistic. And so this is an algorithmically tractable test. Okay, so let me wrap up the talk by talking about a sample application to give you a flavor for the sizes at which we can do this and what happens. There are not so surprising, but I'll tell you. So the data we're looking at is time series of single cell mRNA expression profiles. And what's going on in time that we're looking at? Well, we're looking at cell differentiation. So this is based on a paper by Schiebinger on something called Waddington OT, which looks at developmental trajectories. It's a cool paper that looks at developmental trajectories using optimal transport to do alignment in time. Now, alignment in time is hard, just as a aside, because when you measure the cells, you kill the cell. And so it's not like you can track a cell through time. You have to sort of align them by looking at their position in genome space, as well as possibly in physical space. And anyway, they sampled about 250,000 cells in mouse embryo fibroblasts, which were undergoing reprogramming, which means they dumped something on them, on these cells, that told them to, go to become stem cells and start differentiating. So they triggered a differentiation process, and then they watched it for 18 days. And their measurements were taken at 37 time points. So this is a small data set by the standards of the asymptotics, right? There are only 37 time points. And and at each time point, as you can see, 250,000 total cells. There are thousands of cells at each time point. So that's pretty good. But the number of time points is quite small. And so it's really critical to have enough power to see things in finite samples. And just as a procedural thing, just to tell you what we did, the data was log transformed, and we did a gene selection to take the 2,000 most variable genes. And what we discover is that the tests, the most sensitive of our tests, detect non-stationary behavior. I guess I didn't write this in persistent homology 0 and persistent homology 1. Now, these tests are interesting in persistent homology 0 because looking at cluster structure behavior and that non-stationary there is already a pretty interesting question. And looking at it in PH1 in these contexts where you can interpret what loops mean in terms of developmental processes is also interesting. Okay, so this, these things are developing. You sort of know they're non-stationary, but the interest again was that you can show that these tests work in this kind of regime. Okay, and let me just close by showing you a picture of what this looks like. Oops. So, well, maybe I'll wrap up after the picture. But the picture looks like this. So this is colored on the left. This is colored by time, and on the right, this is colored by cell type. And basically, what you see is sort of everybody starts out green, and then they differentiate. And as they differentiate, they sort of move in genome space, and they get to develop these tendrils. And this was produced by projecting into R2 and doing some kind of force-directed graph. So one comment is that eyeballing this, you would think, well, the shape is changing, but what the shape is doing and sort of what the underlying process is doing in a higher dimension, in the sort of actual space this data lives in, is very unclear. And it's why we need tests like this to tell if these if there are high, because what we're looking for are, just to say something, are higher order dependency structures, right? We're looking for things like dependency structures on the moments of these things, and that's not always maybe often in these contexts not going to be visible to the eye, or you'll see things that are visible to the eye, but you won't really, that'll be an artifact of the dimensionality reduction, not an actual thing about the underlying data. Okay, so wrapping up, sort of the take-home messages from this are that there's this framework that we want to introduce based on functional time series analysis, which is a new and exciting area of the statistics of time series. This provides allows us to give rigorous foundations for statistical analysis of questions about the evolution of shape. And it's a nice framework because it makes it natural to incorporate non-stationary temporal and spatial dependent structures, irregular and noisy sampling, and analysis tools to get rates and non-asymptotic bounds, as well as understanding the asymptotics. And for me, and this is just a function of my personal interest, the sort of application that I want to use this on most is developmental processes in single-cell RNA-seq. Understanding non-stationary behavior, which you expect essentially in any developmental process, but then also detect, in subsequent work, we would be trying to detect breakpoints and information about the dependence structure, which should inform you about the developmental process. Okay, that's it.
Questions for Andrew?
Hi, do you have an interpretation of what the breakpoint is looking at, is detecting when the pH 0 and pH 1 is changing? Are we looking at some sort of bifurcation?
Yeah. Yes, exactly. What you're looking at, in general, no, right? Other than very naive things. In the case of the developmental process, I think what you're seeing is that these cells are, your stem cells are changing into lots of different kinds of, I don't know, here we see skin cells and neurons and, what look like blood cells, and those diverge. And so I think you're detecting that. And in some cases, the developmental trajectories, and this is a little bit complicated to explain the details of when this happens, but there are cases where those things converge again a little bit in genome space, and that's something you might hope to detect with loops.
Sure.
Now, to say there's always the question of, and this is as yet undone, to show that we can use this to detect things that you could not detect some other way. Like, I don't know yet that these tools will have taught the biologists anything they didn't know already about this data set, but that's always the question with topological data analysis.
Aaron?
I'm curious if you've applied this to any more geometric time varying data sets, like, objects that are moving or growing or.
Yes. I should have had a picture in here. I thought I did, or at least a formula. Here we go. Somehow we missed this. So the top here picture is a cobordism, right? Where the topological, if you look at time, is moving across I guess on the, horizontal axis. So you can look at slices like that. And down below, we have sort of circles that are growing, and the growth has a dependency structure indicated by that function gamma tt. And yeah, this works to detect, in these kinds of toy geometric examples, this works to detect the change of the non-stationarity, and the sort of breakpoint variance will detect breakpoints if they're discontinuous changes. So from that perspective, yes, I don't think these are real geometric examples. Obviously, this is sanity check kind of stuff.
Andrew, nice to talk. Do you have p-values for the breakpoints that you've detected in the non-stationarity?
For this genomic data?
Yeah.
Yeah, I think it's like for 8, I can't remember. There are a couple of different test statistics we check, and I think it's at the 5% level for some of them and the 10% level for others, varying from pH 0 and pH 1.
Okay. And I noticed you were using the bottleneck distance. Is there
Yes.
a reason for that? Because the 1 Wasserstein distance would is more sensitive to.
Yes. No, there's no good reason for that. So, but a point well taken, and there's no like you could easily just plug in the Wasserstein distance. There's nothing special.
Okay, thanks. All right, then let's thank Andrew one more time.
Awesome. Thank you.
Thanks, Andrew.